I'd really like to thank the organizers for inviting me to present today, and I'd like to thank you for making the time to attend this presentation. I am going to explore the question as to whether or not animal study workflow software that more closely emulates clinical trial approaches can actually help improve the predictability of clinical outcome. After that, I'll be showing the software that we use as well, and if you're interested in a more detailed look, please feel free to contact me. Be happy to meet with you. So the problem is we have an unacceptably high failure rate in late phase clinical trials. And following clinical trials, we have an abysmal 5% approval rate of anticancer therapeutics. It may be surprising to you to know that this is primarily due to a lack of demonstrated efficacy as opposed to toxicity. So why such poor predictability? Well, it's really simple. Our traditional approaches to how we do efficacy testing and the tools that we use to test them differ significantly from clinical trials. We have significant differences in the study designs, so hodgepodge of various protocols are being run differently with different parameters being run, different sizes, different scopes, different subjects, such as going for more homogeneous subjects genetically, as well as homogeneous diseases that we're testing, so using the xenografts and really not reflecting what is seen in the clinic. And also how we enroll animals, for example, is more on a synchronized basis as opposed to a rolling enrollment as is seen in the clinic. And the analytical reporting methods are not as clinically relevant as they could be. And this is driven primarily due to the limitations in our traditional animal study conduct tools, which unfortunately still in 2015 are Excel-based with a hodgepodge of other analytical approaches such as GraphPad Prism or R or SAS. And we see a great number of labs that are still using paper plus Excel. So effectively, we're using a more manual approach with Excel and these other tools. This leads to significant inconsistencies across labs and even within a single investigator from one study to the next, which prevents cross-study comparisons. There's a great deal of inefficiency because of all the manual steps that are involved, and there's never enough time. Every researcher I know never has enough time, and that's really due to the tools that they're using and the limitations. Data integrity is impacted, mistakes happen. We see errors in recording, in transcription, copying data, and they remain undetected. And for example, we recently encountered one of our customers that asked us to make a report based on one of their Excel macros that they've been using for 10 years, and we found 2 major mistakes in those, and they didn't know for 10 years. We also see scientific misconduct. It is rare, but it does happen. We're hearing more and more about it on the news, and it impacts the integrity of the entire study. One example was a case of, well, I'll just say scientific misconduct that was undetected for a number of years, and it cost the company between $2 and $5 million. So it's rare, but it does happen. And because of how the data is collected in notebooks and Excel spreadsheets, it's usually only available to the originator and to no one else. It remains in these data silos, which are by design unsearchable. If you can't find data, it's effectively discarded, and that is a tremendous waste of data, research, resources, animals, time, money, all of things which are really precious and difficult to come by increasingly. Also, we have a really significant lack of interconnectedness and integration, which limits how we can run our animal studies more like clinical trials. Study conduct tools, by using a common study protocol and methodology under a common software application database, we can improve the methods, so increase subject diversity, disease diversity such as tumor diversity, the methodologies such as enrollment approaches and study sizes, etc. And these can more closely emulate clinical approaches. So the conclusion is, because technology is still very new, it's not yet been proven, but it is highly likely and stands to reason that by more closely emulating clinical approaches with improved technology, that we can improve the clinical outcome predictability. So a new technological approach involves using a common study protocol across multiple researchers or within an organization or within an organization that's worldwide. It really doesn't matter. Instead of having a hodgepodge of various study designs, you have a single study design where you can run multiple studies. If you have limitations at one site, you can run those at other sites as well, and then combine all of those studies and that data under the single protocol. You can employ multiple treatment arms, such as having multiple PDX lines per model. This helps increase the N or the power, and increased power leads to increased predictability or improved predictability. You can thereby piggyback studies, and then we can employ rolling enrollment approaches, much more clinically relevant. We can use randomization, analytical, and reporting methods that are much more clinically similar, and those methods and results can then be saved into a database where they're securely preserved. They're accessible by stakeholders. The information can be distributed, but much more easily. But they're available. They're securely preserved in perpetuity, where they're also available to be searched. So labs don't unnecessarily reproduce studies. We see quite a bit of that. The traditional approach using existing spreadsheets, graphing, and analytical tools has been clearly too complex to manage the studies, reflecting a clinical research workflow. So we have a 5% approval rate of anti-cancer therapeutics. We've been using the same tools and doing it the same way for 30 plus years, yet expecting a different result. So doing it for 30 years or longer, I am the person on the left, in case you don't recognize me. And by virtue of attending this presentation, you are clearly not the people on the right. Study log solution. Our study log animal study workflow software and database makes it feasible for the first time in history to conduct multi-site, multi-arm preclinical trials with randomized rolling enrollment or synchronized enrollment using a common centralized protocol. The application is designed specifically by scientists to automate and facilitate the oversight, and most importantly, to mirror the workflow of animal studies in oncology and multiple disease areas. We have researchers using it in infectious disease such as Ebola, inflammation, neuroscience, toxicology, metabolism, etc., which speaks to the flexibility of the application and the applicability of it. So it is a powerful system allowing you to oversee and conduct studies, knowing exactly what's going on a day-to-day basis. As I said, the software is extremely flexible. It is a hallmark of our software design process. We know that changes always happen on a study. They must happen because you can get to answers much more quickly by being able to analyze in real time as a study is being conducted, and get to answers more quickly, and then you can ask subsequent questions of a study, and so that allows you to do more studies and save on resources, animals, and time, etc. So flexibility is the key. We have a comprehensive scheduling system which allows you to schedule, let's say, asynchronously assigned animals into a study, or different treatments that each have their own schedule. That's very important to know. And for users that are running these studies, they can very easily see what the schedules are when they come in the morning, what is to be done, what's to be done at the end of the day if something was missed, and should a colleague not show up, what duties need to be covered for them. So it makes life a lot easier. It helps lower stress level and improves planning. You can easily align, analyze asynchronously collected data, and we use clinically relevant distribution and randomization methods such as multi-parameter randomization and stratified sampling. These can be done synchronously, or you can randomize on an asynchronous or iterative basis, such as done in the clinic, as you have newcomers or new subjects available to be used on a study. So this is, again, more clinically relevant. We have an audit trail tracking every change in the study so that during the study or long after the study, you know exactly who did what, when, where, and how. We have a system architecture that allows for a single user on a single laptop to use the software for very small labs. We have a lot of small labs using it. Or you can have very large multinational organizations as depicted in this architecture description, where you have a single centralized server and database that synchronizes with, in this case, 3 different sites with their own database, and then multiple laptops and or web portals collecting data or changing the designs or accessing the information, all synchronizing to the database on each site. Software allows for the design of studies. Users can input their randomization methods or distribution methods, the groups, the number of animals, the types of animals, what are the subjects they're using, if they're using wild types and transgenic, all of that information, the housing, bedding, caging conditions, the treatment descriptions, so drugs or treatments or drug combinations. They can also input the disease induction methods, whether that be a PDX or a syngeneic allograft or a chemically induced tumor, as well as they can create or customize their own numerical values that they'd like to collect or measure. So they can be scores or measured values such as net body weight, or let's say serum biomarker levels, or any sort of new numerical value you'd like to collect. Users can view the study, as can stakeholders. Designated personnel such as technicians are allowed to collect data. You have to be authorized to collect data or to be assigned to a study for the specific role. You can change data. If you can collect it, you can propose an amendment, which are amendments are to data are approved, as are the amendments to the study designs. Data can be reviewed, and if the reviewer sees an animal is an outlier, they can actually exclude that data or that single data point or that data type for that animal, or if an animal has been accidentally misdosed and is essentially a protocol deviation, they can exclude the entire animal from all subsequent graphs and analysis. So users can create graphs just a click of a few buttons. They can analyze and create reports and data exports in a variety of formats, all with just a few clicks in a very consistent manner. They can view the study history, again, so you know exactly who did what, when, where, and how, so there is accountability, and this really does help mitigate the chances for any sort of scientific misconduct. Study templates can be employed, so any study can be created and turned into a study template, so you have consistent study designs, or you can also clone or copy a study design very easily to quickly recreate a new study based off an old one and just change the things that are necessary. Studies can then be archived where they're available for search purposes, and they're locked down for editing. They can always be reactivated if needed. And studies can be exported to collaborators that are not part of your database, but they have study log, and then they can import the study and see it as if they ran it in-house. So that's the basis of our study-based functionality. We do have also messaging functionality internal to the software as well. What our customers say about study log? Well, they say a lot of nice things. I'll say that in general. We're very pleased. This is an example from Gensis, and I'm not going to go through all of these, but I will say this: being able to push the data into an ELN makes the lawyers happy. So we do work with your existing LIMS systems. What we see is this is very nice that our software saves them time, resources, and money, and allows them to achieve more results with their existing staff. So what they've been seeing is a doubling or even tripling in the number of studies they can perform, and this is consistent with what we see with researchers at other labs, and this results into a significant net benefit of optimizing their workflow so they can screen the greatest number of drug candidates possible. So this is very powerful. We've been around 13 years. Our core competency is animal study management, and the software, we have a very consultative and collaborative approach, taking all the best ideas and best industry practices and building them into the software and then rolling them out to everybody as they're available. Our customers include numerous academic labs, the National Cancer Institute, 7 out of the top 10 big pharmas, the world's largest academic lab, or the world's largest biotech, and over half the top 20 CROs worldwide. In 2015, we received a BioIT World Best of Show Award by a panel of bioinformatics experts, and we had the distinction of being the first animal-related software to ever receive this award. So we're grateful and honored for this acknowledgement of our approach. So you can contact us at the addresses here. I will be showing the software in the next phase of the presentation, and thank you very much. I have now logged into the desktop version of the StudyLog animal study workflow software. We do have a web version that does everything that this one does except for data collection. Upon logging in, each user sees the studies that are in the database. In this case, all studies are being displayed. That includes all active, all archive studies, which are locked down for editing. It also includes, if I expand this, the studies that are still listed as private, so I don't see those. Only the people that are assigned to those studies see them. Studies in various stages of development, such as draft, waiting for approval, or not started. And I can also see the active studies. And these are all the studies that are currently ongoing. And but I might not want to see all those studies. I might just want to see the studies that I'm playing a role on or I've been assigned to. So I can click on the button that says I am assigned to, and I'll see just a list of the studies that I have a role on. So there, that list is made a little bit shorter. And we also have the option by, you see the little gold stars on the left. When those are checked, that makes a study a favorite study of yours. So if you're a stakeholder within an organization, such as a project manager or upper level management, or even somebody that's essentially principal investigator, and they have several people working on a variety of studies, and they just want to keep tabs on their favorite studies or the studies that maybe are more meaningful to them or most important to them, they can very easily just take a look at those studies. And it's very easy to simply highlight a study, right-click on a study, and create a graph. So if I right-click on this, we have the options to create survival graphs or other graphs. It really doesn't matter, so I'll just show you an example here of a survival graph. This is for a colitis study. As I mentioned, StudyLog is used not just for oncology, but it's also used for a variety of other study types. And I can just actually very easily copy the image to a clipboard and paste that into a Word or PowerPoint or Excel document or my email and distribute that information that way. And this makes it very easy. You can also use this as part of any reports you'd like to generate. Although our Excel exports and other reports create their own graphs, so depending on what you need, that graph may be just generated automatically. But this allows you to very quickly to generate a graph and view that literally hot off the mouse. So I'll close this graph. And I'll open up another graph. And this is a graph of a dose response study, and what I see are the group means of both body weights and tumor volumes. And we have the options where we can actually show the number of animals that power each time point, so you've got an idea as to where you might want to do your statistical comparison or statistical analyses. You can actually show the values. You can, as you see here, we've got the show censored data box checked. So as I mentioned before, you can censor or exclude data. And by checking this box, any data that I've censored will be applied to this graph. So in other words, we can exclude any data that we want to exclude from this graph or these graphs and any of the reports. We have also the option to graph not just absolute data, or values, but relative values, the delta values, group means plus or minus standard error with error bars or not, geometric means, medians, etc. So they're all available here to be graphed. And if there are dosing has happened here, you'll see dosing day arrows. Now in this case, you see I've switched this to a relative graph, and if I'd like to just basically hide all this information, I can simply hide it. Excuse me. And now I can paste that into my report if I'd like to do that. So I'll go back to all of the list of all active studies. And something that's really important to be able to do is to find studies. So we're always looking for information, want to know what's going on any given day. You see we have a variety of columns within this view or screen. We can move any of the columns from one place to another. I want to move articles over here, I can do that. If I'd like to move the cells over on this side, I can do that, or move it back. If I want to get rid of a column entirely so it's not displayed, I can customize my view that way and just do it like that. So there's a lot of flexibility here, and that applies to all of the study designs. So I'll move that articles back in here. And now, what I'd like to do is show you how we can find information. So if I want to look for a study with a certain, let's just say, cell line, I could type in, let's see, colo, and I'll see all the colorectal ones. Okay, there's only one study there. But there's other ways of searching for studies, and we can use our Boolean search tool. So if I want to type in a search term here, such as, just see here, HER2. The system will look through the entire database and bring up all the studies that have HER2 used on them. So if I want to just simply highlight them and then generate a report, I can do that. So I've got a bioluminescence study, dose response study, and some others. In this case, I'm going to highlight my dose response study and generate some reports. So I can show you how we can very quickly get to the results and generate some analyses. The first report I have, well, I've got a wide variety of reports that I can choose from. In this case, I'll just run the preclinical research report, which is essentially a protocol report. It has a smaller number of options as to the format of the report and choices. Each report has its own, and also we have a variety of report formats based on different disease areas and model types, such as behavioral science has a different type of Excel export than, say, an oncology report. So in this case, we see a more formalized protocol report, protocol and report output. This is for a contract lab customer that we created this for. So you've got your table of contents, and then details about the design of the study, the objectives. So all this information that has been entered into the design of the study gets pushed out here. The design and methods, the test articles, details about the treatment groups, the frequency of administration of each, and if we see all the tasks that have been scheduled for the study. And if we scroll down here, we see some graphs. So we see a group mean body weight graph, as well as a group mean tumor volume graph. And then down below that, we see actual raw data, so the length and the width and the calculated tumor volumes along with the body weights. So that's just one example of one report that we can create. I'll show you a few more, not to go into too much detail. One of our more popular reports is the Oncology Report. And in this case, I can choose to include the unassigned animals and as well as maintain the excluded data or censored animals, and they'll remain excluded. And from this report. That will generate shortly. So here we see the oncology report. If I click on the tumor volume worksheet, we see a graph of the group mean tumor volumes along with tables of the group mean values and standard errors. We see graphs and tables for percent T over C tumor volume, percent inhibition, percent delta T over delta C, and mean percent delta inhibition. There are other outputs or other worksheets here. So for example, if you want to copy and paste data into GraphPad, you can do that in that format. However, we do have the ability to simply right-click on a study and push that data into a GraphPad format that you've provided us. So if you have a template that you use routinely, you can send it to us. We can include that into the software itself. And you can see the same sort of worksheets with group mean, body weight graphs and tables, etc. So again, just one report. I'll show you another example is our consolidated report. So this is for the same study. This is showing group mean tumor volumes as well as relative tumor volume, so percent change from initial with the tables. And again, tables for mean, percent inhibition, medians, etc. And if we'd like to actually see more of the raw data side of things, I could see the individual tumor weights, the individual tumor volumes. So what I'm seeing down here on the bottom is I'm seeing the raw data, so length, width, and height, although you could do length and width and calculate the volume that way. So any formula you use can be applied. But in this case, we've got individual group graphs for absolute values as well as log values. And then here we see a table of the calculated tumor volumes along with the animal IDs and the fates. And if I'd like to point out on the left here, we have two sets of IDs, temporary IDs and permanent animal IDs. In this case, the temp IDs appear to be microchip IDs. So that is not a problem to work with microchips. The system just loves it. So I have also run a group comparison report. So I will say this, not everybody likes to do statistics, and you don't have to know much about it. All you have to know is how to actually generate it. And in this case, the system determined whether the data was parametric or non-parametric on each of these study days. And as we see here, because we have an ANOVA, the data is parametric. And the p-value, which is highlighted in bold, is lower than 0.05. So that tells us there's likely to be a difference between at least two groups. We don't know which groups until we look at the post hoc test. In this case, we see there's a difference between groups 2 and groups 2 and 4 and 2 and 1 in terms of body weight. And the post hoc tests also have results. So other post hoc tests, the Tukey's and Chaffee's. So we could do this for body weight or for any data type. And they're very easy to generate, and a lot of people appreciate not having to actually know a whole lot about statistics. If you do love statistics, don't worry, we do have the ability to push the data out into a variety of formats. If you give us an algorithm, we can actually create an output that runs that algorithm. As long as you've got R installed in-house, we can run it and then bring that data back in like we do with our multi-parameter randomization method. Or we can just do it using a report. So a lot of options. Another report option we have here, I won't show you, we have clinical observations, a variety of data exports, protocol exports, formulation reports, group data, raw data reports, and then here you see reports that are specific for different neuroscience models, etc. So really as a testament to the flexibility of the software that we've got such a wide variety of outputs. And of course, as I mentioned before, we've got the ability to let users create their own templates using blocks of each of these reports and compile their own custom report.
So that's all I'm going to talk about reporting. If you'd like to see more and learn more about this, please let me know. Contact us for a demo, and we can show you a lot more. So one thing I'd like to show you is our today's tasks. So that's under the menu, and what it does is for each user, based on the login, it knows who you are, and it brings up all the studies and the tasks that are scheduled for today. So in other words, it's your to-do list for today, so which is nice to come in the morning and see what you've got going on. So I see on this study, number 16, I've got serum to collect from 65 animals. On this imaging study using IVIS, I've got body weight, flux values to measure, as well as luciferin to inject. I've got a growth curve where I've got body weight and tumor volume for a number of animals, and I've got a drug combination study where I have to measure both body weights and tumor volumes, as well as do a number of dosing to a variety of groups. And on this top study, this dose response study, I've only got dosing to do, and that's scheduled for specific times. So we see from 6 to 11 a.m. And you can assign tasks to specific individuals, so it would show by which individual that they're assigned to. So I'm only seeing things assigned to me for today. I could see other people's if I click on show tasks assigned to other users, and I could see the entire week as well. So I know what's going on, not just today, but tomorrow and the rest of the week. And this is also useful to check at the end of the day for most researchers so that they know exactly what they might have missed that day. So everybody gets busy running multiple studies, so it's nice to have a place to check it and see, okay, did I get it all? And you see the number of tasks that have been done on the right here under the progress bar. The 0 out of 10, 0 out of 10 tells us none of the doses have been done. But once you see 10 out of 10, you know that they're all been done. So it's really a helpful tool for oversight, as well as the users themselves that are running studies, or the technicians, to actually manage what their schedule is for the day. So we can highlight any of these studies and just simply open them up. I'm going to open up the efficacy study. In this case, it's an IVIS study doing some bioluminescence, measuring flux values. So we have fixed doses of luciferin. So we can start with our first animal. We can do animal one by one or group by group, or I simply just highlight and hit the space bar and record that I've actually injected the luciferin. And if I like, what I can do is right click and apply that to the entire group or all the animals, and they'll all have identical date stamp, time stamp, and user stamps, because every measurement is time stamped and user stamped down to the second. So these would all be identical, which would kind of make sense. You dose them, and then you can go ahead and run your animals using your IVIS system. So in this case, I can select an Excel file that has got some data from some flux data. So I will highlight that. Copy it, go into study log, highlight those fields for those animals. Oh, wrong ones. The ones with the little clocks tell us that those are scheduled. So that's how I know what I have to do today. So in this case, I paste in those values. They're there. And if I would like to actually associate or attach a hyperlink, this is a linked field. So I can actually select a file, an image in this case, the original image, and right-click on it, and I could apply this image to a number of animals. So I know there's 5 animals in this image, so I'll do group 1 and a couple of other animals that are in that image itself. Okay, so I've applied that to multiple animals, and if I want to see the image very quickly, I can just double-click on it, and there I see it. So it's there associated with the values, and if we'd like to associate it with comments, we could do that as well. And I can select body weights, And as I mentioned before, we can create dictionaries that are specific to data types. So we can link files, but we can also show comments. So in this case, if I weigh an animal and it's extremely low body weight, we'll say 15 grams, there might be an explanation for that. So I can right-click and simply say, okay, well, it's not due to anything that happened other than it's got a malocclusion. So that's important to know if you're looking for toxicity or dose response. The animal's not losing weight due to the treatment, but actually due to a physical problem. So this is just one example of data collection. If we wanted to sacrifice that animal, I could just very easily right-click and enter in a mortality observation. And instead of recording it as alive, I can just say, okay, we're going to do a euthanasia. Let's just say, it could be simply removed from study. And there we go. You see we've got a variety of other mortalities. For example, animals that have excessive tumor burden or body weight loss, so you can record that as well. If I have another animal that's, let's say, lost a lot of body weight, and I don't have a threshold limit being reached here, but I just know that that's unacceptable, so I could just basically enter in a mortality observation for it and record that it's excessive body weight loss. So now this, I'm just calculating in my head, but on another study, I can actually see where I've got previous data. And you can see here, we've got tumor volumes in red here that indicate that the animal previously has exceeded tumor volumes that are acceptable or that the animals need to be monitored more closely. So for example, if I want to body weight an animal and put in 25 grams, it'll calculate the dose. I record the dose. In this case, it's not a fixed dose. It's actually based on the actual body weight. So the system will calculate the dose volume based on the most recent body weight. So I could put in a value here of 12 for the length, and then I'll put in 13 for the width, which is a mistake by definition because it would square that. So our system corrects that mistake, and if I have a tumor volume that's very large and I've exceeded the threshold limit, which I seem to need to really be able to exceed that. Let's just try something here. So there we've exceeded the threshold limit, which in this case is 2,500, and it tells me that information. So in this case, I can right-click and enter in a mortality and record that as due to tumor volume exceeding the tumor volume threshold. And if I'd like to, again, put any comments about a tumor volume, I can do that as well. There would be a different comment menu. So in this case, it would simply be right-click, and then in this case, necrotic, necrotic mild, necrotic severe, or we'll just say ulcerative moderate. Okay. So these are just some of the things you can do. I can enter in clinical observations. So I can collect data for all tasks that are scheduled, but I'm not forced to do that. I can also have the freedom and flexibility to do tasks on the scheduled days or to choose to do them on other days. So in this case, I might just want to enter in clinical observations right now. So I just select that as a data type, and I will double click on an animal. I can say, okay, this animal happens to be normal, but this animal is not normal. And so I'm going to call up and add in a clinical observation. So we have a clinical observation dictionary that comes with the system. It can be modified, as I mentioned before. Users can create the dictionary superheadings, such as locomotor, and then see all of the specific details or observations that are related to that locomotion. So here we see catalepsy, ataxia, etc. If we look under respiration, we see respiration-related observations. So if I say, okay, tachypnea and I click Add, when I set up the dictionary, I required that a severity be recorded. So in this case, we'll say moderate tachypnea, and then that would be recorded. Now, alternatively, I could remove this and do something else. I could record things that are maybe not clinical observations. I could actually record things like histology descriptions, or let's just say the colon is severely enlarged. So I could say, okay, colon, specify enlarged, and I could specify a size or a score depending on how I want to set up the dictionary. So in this case, maybe we'll just say a score of 4, and we'll just say at the distal end, we'll say the posterior end, that's fine. I'll just click Add, and then that's in there and recorded. Should we hit OK and then save? But in this case, I don't want to record it because I want to keep showing you other functionality within this tool, and that is specifically, this is very much appreciated by researchers, and that is the ability to record where they're collecting tissue, and how they're collecting tissue. So if we're supposed to collect tumors and cut half of them and formalin fix them, I could put it in a cabinet. And then specify the location, the rack, the box, add that. If I'm cutting that tumor in half and putting the other half into a formalin fixed block, that's going to go into a cassette, into a freezer, shelf 1, freezer 1, shelf 3, rack 2, box 2, and click add. And then what I can do is click OK, and then I can click out and then right click back on here. And I can then apply that to all animals. And so then I can run a report and always know where those tissues are stored and located. And if I change the location, I can change the location, or if I create new samples, such as slides, I can record the location of those slides as well. So that's kind of the idea around data collection. You can collect any data type that you want, whether it's scheduled or not. You can collect it as you need to. You can select what you want to do when, and the system will record exactly what you did, whether it was planned or not planned. It doesn't matter. It will be in the database. Now, if I want to change something, I can change something, but if I have the auto lock cells box checked, you see here it's checked, I can't change any of that data. If I uncheck it, now I can actually go through and change this value. So instead of maybe 0.25, I reweighed the animal, maybe it's 0.26. So that's an example of ability to very easily change data as you need to. Once I hit save, that data will then be committed to the database, and then to change it, I need to actually do a formal data amendment. Okay, so if I close my computer or my battery runs dead, the data will be parked in the database where it's preserved, but it won't be committed to the database. So you have to hit save to actually commit it. So we do have a tool to review data and amend data, and that's here, available also from the review data function. So I could actually do a very quick full expand on this data. So I could take any single data point and highlight it, and if I want to exclude it, this is how or censor it, I could just basically click turn it off. Or if it's not just the body weight, let's just say it's body weight and tumor volume, I could do that as well. And I'll highlight the tumor volume, and I'll exclude that. And I'm going to exclude all the tumor volumes, let's say due to a necrotic tumor. Or maybe I want to exclude the entire animal and turn it off, basically censor all of the data because I misdosed it. So I could turn that animal off entirely, or exclude all that data in one step, and then in all the graphs and reports it will be excluded there as well. If I need to align asynchronously collected data, you see here the align data by, those are tools by which I can do that. I can shift data. If I need to amend a data point, it's very easy. I can just simply click highlight and amend the value. and put in the changes along with my reasons. And if I have the appropriate authority level, I can approve my own data amendments. If I'm a lab technician, I don't have that authority, I can propose the amendment to the data. All previous versions of the data will be kept in the system, so you always know what the data was. If you change it 6 times, all 7 versions will be there. So if I put in 20, that will get changed, recalculated, and I can approve it, and then it's saved into the system. And all data amendments are accessible and also available in a variety of different reports. So I can choose to save this data, or if I want to discard it, I also have that flexibility. If I made a mistake and measured a bunch of animals with my calipers and maybe they weren't zeroed out, or my balance wasn't zeroed out, and I don't want to go back and delete and rewrite the data, I can just basically discard it in one action and just click discard, and then that data will be excluded. And now I can close. I should mention that we do have the option to show previous data while we're collecting data. So you have reference data, so for example, previous clinical observations or tumor volumes. If that's important to you to look at while you're collecting data, you can actually have that available for viewing. So going back into the design of the study, I can highlight any study that I want to take a look at, and we'll just say, okay, let's just take a look at the study. So I'll highlight this study, the dose response. I'm going to go into edit mode and just very briefly just show you the highlights of the design, and then we'll wrap it up there and do some questions. So each study is represented by a series of tabs. So the first tab is the general tab. You can put in your study number or name, whatever you want to call it. The system can automatically generate study numbers. Each of the fields here, you can free text and type in if you want, or just select from what's in the drop-down menu. You can lock these drop-down menus so users only select from what's available. You can also make fields required so that they get a reminder every time they go into edit mode. So you've got a lot of flexibility. It can be as loose as you want or as tight as you want. We recommend like making your IACUC protocol, your animal welfare license, a required field. That's a good idea, just good practice. You can put in your title, objectives. At the end of the study, you can put in your results and conclusions, which are here, which, since I don't think we finished this study, I don't think we have any results and conclusions. So no, we don't. But if we did, they would appear in any of the reports. You put in your dates as best you can, as you know them. Obviously, you don't necessarily know when dosing will begin until you're ready to randomize. And then when you do randomize, basically you just put in the dosing begin date. And specify if that's your day 0, if you'd rather have your inoculation date be your day 0, however you like. So our personnel tab allows you to assign personnel to a study. So I can add individual users. So I can add a technician, for example, Bob from work group 1 as a study technician. And maybe I'm going to add, get my mouse to work here, can then add a department or a group of people as technicians, so a team of people. We'll just say work group 2. And all of the people that have the technician role or responsible technician, in this case 4 out of the 5 people in that group have that role or permission in their profile, and so they can actually collect data on the study, review the study design, or rather view the study design. They can run reports, but they can't change the design of the study. So only authorized people can do that, which includes the PI or the study director and the study coordinator. So study reviewers can just look at the data. They can't actually collect any data. So you have complete flexibility as an organization to add new roles, which like these, and then define what the permissions are for each. And here's where we can make a study private. And if you're going to want to assign people to approve the study design, you can check them here, and they'll get messages to approve the design or not. And it will be active once they've approved it. So we can assign the animals that are going to be used on the study. I'll have all the details about them, the vendor that they come from, the species, and then we've got vendor and species-specific strain names because that nomenclature is important to have consistency for. Your euthanasia method, here you see they're identified with microchips. We see the 2 sets of IDs. If there's any fate or threshold limits that have been exceeded, you would see those here. In this case, all the animals are alive and happy. We can bring in animals from previous studies that are left over, let's say unassigned animals, and we want to reuse them for another study. You can do that. We can print cage cards from here. I'm not going to do that. We can also print group cage cards as well. We've got a variety of formats that we can provide you as part of it, but we can also customize cage card formats. Groups are very easy to create and add. You can have any number of animals per group and fill in all the details you like about the bedding, caging, the housing conditions, things that qualify these animals as part of the study. So no limit to the number of groups here. We also have our treatments or our test articles. In this case, this is a dose response study. So it's one vehicle control, as you see, administered intravenously with the dosing rule being, or dose volume being 10 mils per kilogram based on body weight. And as you see the test article, we have 3 different drugs or dose level, or single drug with 3 different dose levels, also administered by the IP route, but these are described as test articles as opposed to vehicles, vehicle controls, or negative controls, or let's say untreated controls. We get a whole variety of different descriptors for treatments. And the system will also calculate formulation information if you choose to enter in that information. That makes that process significantly easier. We also record the disease induction method, which for oncology studies is usually going to be a xenograph or a PDX and the specific cell line and number of cells and the volume and that sort of thing. It'll also calculate how much of each of the components that you need. For example, a 1-to-1 matrigel to HBSS ratio. It'll calculate how much volume you need of each, and you record the location. And pertinent information that 5 years down the road you might not remember, or maybe you forgot to record, but in this case it makes it easy because especially if you're cloning a study, this information will be here. But you get details about the inoculation as well as preparation. So what kind of media you use, the confluence, etc. Any sort of samples that you set up to collect on a study can be defined here, and this makes it very easy because you can basically view and specify the details. I didn't show it in the data collection, but in the data collection when a sample is to be collected, it actually shows these instructions right next to the checkbox, like 100 microliters are to be of blood are to be collected in a red top serum separated tube under isoflurane anesthesia. And of course we can also very easily use our print sample label generator to create samples that we can very easily print out, in this case for plasma with all the group IDs and animal IDs accordingly. And moving on, one of the most important pages or tabs in the system is the tasks and measurements. This is where you can set up threshold limits for body weight, so 20% or 10% body weight loss, your tumor volume information, details about tumor weight. It'll calculate net body weight by determining what the weight of the tumor is and then subtracting that from the actual body weight. So if you have threshold limits based on that, that can help you more closely monitor the health of the animal. So this is where you would add all sorts of tasks. We have a variety of tasks that you can enter in, but basically any numerical value you want to enter in, whether it's a tumor volume or a food consumption or water consumption, or a custom value like a serum biomarker or a tissue weight, necropsy tasks, sample collection tasks, temperature, etc., etc. And the standard tasks that we have in the system already, as I mentioned, are here, and so you can just simply select those if you don't have them already. So in this case, I could select, okay, let's put in the number of metastases, and that will then be a data type that I can then record and collect. And so we've got the units, number of mets, and there it is. So we've added that to the study. And then we could set up schedules and view them accordingly. So any task can have a schedule. Usually the schedules differ from task to task, so dosing is usually going to vary relative to, say, body weight and tumor volume measurements. So bring my schedule up here. Or it could be that I haven't scheduled anything. So, oh, I have. Okay, so here we go. So what I see is a lot of time tasks as well as body weight and tumor volumes over time. I could show study days if I like, or I can show this schedule in a month view or a week view. So we see have some time tasks. So if I see the work week view, I can see it that way, or a day view. So a single day. So for today, from 6 a.m. to 11 a.m., I've got dosing of these drugs right here. And the time non-specific tasks, such as body weight and tumor volume, are not scheduled today. I've only got dosing scheduled today. So that is basically the details of what we collect in terms of information on a study design. You've seen how that then translates into creating schedules for people, as well as those schedules then showing up as scheduled tasks in the data collection screen. Collecting all that data then allows you to very easily generate reports and graphs within the system, and that data is then preserved into the database. So that is my presentation. If you have any questions, I'd love to hear from you, and thanks very much for your attention.
Okay, I'd like to answer some of the questions that we have had posed here. So one of the questions that we have is which program can be used to build graphics on big data? Well, I will say this, that we have internally a graphic program, as I displayed within the application, as well as the ability to pull data in from multiple studies and apply that on the same graph, both internally and in our multiple report formats. We can push data out into a variety of statistical analysis and graphing programs, such as R and Prism and SAS if you need that as well. So we do have that ability. So that was a question from Aline Gomez. If there's any other questions, I'd love to hear them. And it doesn't look like I'm getting any additional questions right now, but I would like to say that everyone has different needs within the Neurome Lab, whether it's large studies or lots of small single-day studies or long studies, but the software is applicable to labs of every size, and the time saving is, of course, proportional to the extent and amount that the software is actually used. So the time savings are significant, and everyone is usually curious to see how does the software apply to my own personal situation or my own lab's workflow and the kinds of studies that they're running. And my answer to that is everyone is different. We have endeavored to build the software in as flexible a way as possible so that it is applicable to a variety of study types as well as disease areas. And we'd be more than happy to show you on a one-on-one web demo to actually with even examples from your own study types if you want to send those to us in advance, and we could set that up for an individualized web demo and walk you through the software and see how it works in your particular situation. So it doesn't look like I'm getting any more questions. Does anybody like to type in a question? Okay. Again, you could reach me at eibsen@studylog.com, or you can find us on the web at studylog.com, and we'll be more than happy to set up a demo over the web at a time and date of your convenience. So With that being said, I'd like to thank everyone very much for their time and attention and their interest, and hope we have the opportunity to meet with you and maybe discuss any additional questions you might have. So thanks very much, and I wish you all a good rest of your day.